Pseudo-coelomates. We are discussing about pseudo-coelomates. Pseudo-coelomates. Body cavity is in some animals. Body cavity in some animals. Okay, some animals. Body cavity, not body cavity in some animals. Body cavity is not fully lined. Okay, not fully lined. This one you have to remember. Not fully lined. Okay, by the mesodermal epithelium. By the mesodermal epithelium. Okay, by mesodermal epithelium. Okay, but the mesoderm is formed as scattered pouches. Very important. Scattered pouches. Okay, this one you have to remember. Scattered pouches between the ectoderm and endoderm. Between the ectoderm and endoderm. So this is the one. Such body cavity is called. Okay, such body cavity is called, what I said, pseudocoel. What do you call that one? Pseudocoel. And the animals which have the pseudocoel are called pseudo-coelomates. Okay, and this fluid, this is pseudocoel is filled with the fluid that is called pseudo-coelomic fluid. Pseudo-coelomic fluid. This is the point you should have to remember. So the next point what should have to remember in this is best example for pseudo-coelomates. Examples are roundworms. Best examples, roundworms. Roundworms. Okay. Then this pseudo-coelomic fluid. Pseudo-coelomic fluid acts as, the pseudo-coelomic fluid acts as hydro. Okay, very important. Hydrostatic skeleton. Okay, the pseudo-coelomic fluid acts as a hydrostatic skeleton and allows free movement. What is that? Free movement of visceral organs. Okay, visceral organs. These points you should have to remember. Free movement of visceral organs, and for what? Circulation. Okay, circulation of nutrients. For example, here I am telling, see, this is ectoderm, this is endoderm. So here the meso pouches, scattered pouches. These scattered pouches only called between the ectoderm and endoderm. This is only called mesoderm. So this kind of not perfect mesoderm. These are mesoderm scattered pouches. Okay, this kind of only called, this kind of coelom is only called, what do you call? Pseudo-coelom. Okay, pseudo-coelom. This point we should have to remember. This is ectoderm, this is ecto, but this is endo. So middle one without any, what do you call, perfect membrane, just mesodermal pouches, scattered pouches are there. Here scattered, here, this is called mesoderm. This kind of cavity is there, in the sense they are called pseudo-coelomates. The fluid, here the fluid is there. This fluid is called pseudo-coelomic fluid, and this pseudo-coelomic fluid acts as a hydrostatic skeleton, allows the free movement of visceral organs for circulation and nutrients. Okay, children, the next one we are going to discuss about eucoelomates. What is called? Eucoelomates. the eucoelomates. Remember, next one, eucoelom are also called true coelomates. Eucoelom or true coelom. It is a fluid-filled cavity. Fluid-filled cavity. This is the first word you have to remember. So it is developed in the, where it will be, develops in the mesoderm. Develops in the which part? Mesoderm. It develops in the mesoderm. And this mesoderm covered by epithelium. This mesoderm is covered by epithelium. That epithelium is also called, what do you call that is? Peritoneum. What do you call? Peritoneum. Okay. So these are like this true coelom is there or not? That's why these are called eucoelomates. What do you call? Eucoelomates. So based on next one, next category, eucoelomates. So based on what you called mode of the formation, based on the mode of formation, based on the mode of formation, okay, of coelom, based on the mode of formation of coelom, the eucoelomates, these eucoelomates, what you call eucoelomates are classified into two categories, classified into two types. Okay, this one you have to remember, two types. Number one, what is the number one? The first one is what you called cytocoelomates. Cytocoelomates. Okay, what is the meaning of cytocoelomates? So, in these animals, cytocoelomates, in these animals, okay, in these animals, what will happen? In these animals, the body cavity is formed by body cavity. Okay, body cavity is formed by splitting of mesoderm. For example, here I am telling, see, examples: annelids, okay, annelids and arthropods, annelids, arthropods and mollusks. Okay, mollusks. Okay, this kind of next one, cytocoelomates. Next one, eucoelomates. Okay, or sorry, enterocoelomates. Enterocoelomate animals. Enterocoelomate animals. What are these? Here the body cavity is formed from where the body cavity is here. The body cavity is formed from splitting of mesoderm. But here the body cavity formed from, you know very well, formed from the what you called mesodermal pouches. Okay, mesodermal pouches of, here the word you should have to remember, archenteron. So guts lesion, archenteron. Okay, what is archenteron? That gut region, that what you called endoderm region. Okay, examples. What are the examples? Echinoderms. What you called echinoderms and hemichordates. Echinoderms, hemichordates to chordates. Okay, hemichordates to chordates. This one you have to remember. Okay, chordates. So what I'm telling, Eucoelom or trucoelom, fluid filled cavity, completely develops in the mesoderm. So suppose this is ectoderm, endoderm. This is a complete, what do you call, complete fluid filled cavity. So this is only called, what do you call, the eucoelom. So it is formed from, this is called what? Mesoderm. This mesoderm is completely surrounded by epithelium that is called peritoneum. So eucoelomates these are called eucoelomates. Based on the mode of formation of coeloms, eucoelomates are classified into two types: cytocoeloms. In these animals, body cavity formed splitting of mesoderm. Examples: annelids and arthropods and mollusks. Enterocoelomates, animals' body cavity formed from the mesodermal pouches of archenteron. Examples: echinoderms, hemichordates to chordates. Okay, so picture I am drawing. With the picture, I will show the what is cytocoelom, what is enterocoelom. We will discuss. Yes, cytocoelom. Development of cytocoelom structure we are going to discuss. See now, this is the, for example, this is the gut region. Very important. Okay. So now from this area I am drawing like this. So here what is there? This is the thing you should have to remember. What are these? Okay, this one. So this one you should have to. This one you should have to remember like this. Okay, like this or there in the sense. This is called first one. This is called endoderm, right? This endoderm. What is this? This is called ectoderm. What about this? These are only very important. These are early, what do you call? Early mesodermal cells. Early mesodermal cells. What they will be? This next stage what? The next stage, what will happen? As it is, I am drawing like this. Okay, the circle. Here I am drawing like this. And what is there? That like this. Then what is there here? You observe carefully. So that will be like this. Okay, so this is the most important. So this one is called, the middle one is called gut. And what about this? This one will be called split in the, what you called, mesoderm. Split in the mesoderm. The next one later it will be developed into what you call, you see, observe very carefully. Okay, so now that gut is as it is, now it will be complete. Okay, complete formation. This is only, this particular structure only called what you called coelom. Okay, so what you called developing the coelom. What you called developing coelom. Developing or developed coelom. This particular part. So if you label what is this? Ectoderm. Ectoderm. Come on. This particular part, what is it? Gut means you know very well. Endoderm. And this one, this part, this is only called mesoderm. Mesoderm only contains what you call the coelom. The coelom is filled with coelomic fluids. So this is about development of cytocoelom. The next one, development of enterocoelom. Here mesodermal cells to complete what is called coelom formation we identified. The next one, what we have to discuss is what have to discuss. This is the point you have to. The next one is development of enterocoelom. Development of entero. What do you called? Coelom. Enterocoelom. See now, here the diagram will be like this as it is. Okay. But here the what is additionally what should have to remember. So what we have to remember is these are the cytocoelom, what we call enterocoelom. Okay, this is early mesodermal cells. Early mesodermal cells. Early mesodermal cells. And what is this? Ectoderm. And what is this? Endoderm. Okay, these points you should have to remember. So this is archentron. Okay, later what will happen, you see the next study, what will happen, you see the round circle as it is means you know very well ectoderm and gut region. So these are developing, you see these are developing like this. So now what are this? This is what do you call this is called gut. What are these? These are called separation. What do you call? Separation of pouches. Separation of pouches from the gut. The next stage, the next picture, if you see, come on, observe very carefully. So the next picture, if you see, what will happen? The coelom will be developed. So the coelom development will be like this. Okay, coelom will be developed. So what is this? This is called developing or developed coelom. So from where? From where it is? From here. From here. Archenteron. Okay. They are the archenteron mesodermal pouches. These are mesodermal pouches of archenteron. So these are converting into coelom in the sense that is called enterocoelom. What do you call those are? Enterocoelom. Okay. Enterocoelom. Next one. The next concept is segmentation and notochord. The next concept. The next concept is segmentation and notochord. Here I am writing segmentation. What is segmentation and notochord? Very simple. You should have to remember. Okay. In some animals, the body is some okay, some animals body is what body is externally and internally divided into a series of what you call repeated units. Series of repeated units called what you call, those all are segments, called segments. Okay, with a serial repetition, okay, with a serial repetition of some organs. Repetition. Okay, repetition of some organs. Okay, that is only called. So serial repetitions is there in the sense what we will be called that are metamerism. What do you call? Metamerism. So next one, the simplest form metamerism. Okay, simplest form. Okay, this is a very important. The simplest form of segmentation, where we can see the simplest form of segmentation, where you can see is the simplest form of segmentation found in annelids, where you know earthworms. Okay, annelids. Okay, And in annelids, you know, in annelids, what will happen in annelids? What will happen in which each unit of the body is in which, okay, each unit of the body, okay, in each unit of the body is very similar to, is very similar to next one, very similar to next one. Okay, but in arthropods, but in arthropods, you know very well, arthropods means what is that? Cockroaches. But in arthropods, okay, the, what is that? Segments may look different. Segments may look different, so for example, here I am drawing the diagram. So different, and functions also are different. Functions are also, what we are going to see, are different. For example, in earthworm, so for example, you see earthworm will be like this. The earthworm, okay, first segment, second segment, third segment, fourth segment, fifth segment, sixth segment, 1 2 3 4 5 6 repeated segments similar. So this is similar, this is similar to this is same, all our segments are similar, that is called metamerism. So this is earthworm, all segments are similar and they have same functions like that. But in the cockroach, you see this is like this, and what is that, like this and like this. Okay, so like this, what is that, is the pro head, what do you call, thorax. Okay, this is abdomen. So what you are understanding from this, what you now the understanding is, the segments are different shapes, different structures, so and different functions. So that's why here mentioned cockroach segments may look different, so different, and functions also different. But earthworms all are very similar to the next one. This is similar to this one, similar to this one, similar to this one. That is why it is called annelids. Okay, the next one, segmentation, is also called, what is called, metamerism. The next concept, what is the next concept is, what is the next concept, that is called notochord. 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 If notochord is there, notochord is present any stage, any stage. Notochord present in the any stage of their development. Animals, okay, animals. Here I am defining completely animals, which consists of, okay, which possess notochord, possess notochord, possess notochord at any stage, possess notochord at any stage. Okay, this one you have to remember, at any stage, possess notochord at any stage of their development, of their development. This point you should have to remember, of their development, are called, what do you call, these all are called chordates. Okay, for example, here I am telling chordates. So from where this notochord is forming, notochord is forming from which part, you know, mesoderm. Germinals is there or not, mesoderm, and it will be, okay, it will be rod like structure. Notochord is which like structure, rod like structure, rod like structure. Next one, it is formed, it is formed, this point you should have to remember, it is formed, okay, it is formed on the dorsal side, on the dorsal side. Okay, dorsal means upside, dorsal side, during, what you call, during embryonic, what you call, embryonic development. This point you should have to remember. So during embryonic development. So based on the presence or absence of notochord, if notochord is present, if notochord present, okay, what we call those animals. If notochord is present, if notochord is present, those animals are called, those animals, what do you call those animals are called, very simple, what do you call, no chordates. So what do you call chordates. If notochord is present, they will be called chordates. So what are the chordates? Examples I'm writing. Cephalochordates, cephalochordates, two cephalochordates, urochordates, okay, cephalochordates, urochordates, pieces, what we call pieces, and another one is mammalia, pieces and mammalia. These all are called chordates. Next, if the notochord is absent, if the notochord is absent, if notochord is absent, these are called, what do you call, those all are called, what do you call, if the notochord is absent, okay. So invertebrates, what do you called, invertebrates are non-chordates, what do you call, non-chordates, okay, like that you can remember. Absence of presence of notochord is chordates, absence of non-chordates, what are the non-chordates, come on, non-chordates, these are the important non-chordates. Examples, which one, porifera 2, what is called, porifera 2 hemichordata, porifera 2 hemichordata, like this you have to remember. So what is that one, what we discuss, children? Notochord. The animals possess, what do you call, the animals possess notochord, rod like structure, or any stage of their development are called, what do you call, any stage, either larval stage or adult stage, they have notochord is there, in the sense that kind of, what do you call, notochord is there, in the sense they will be called chordates. Notochord is originating from which part, mesoderm. It's a rod-like structure. So this is originated from which layer, it seems? Germ layer, mesoderm. It is a rod-like structure. So it is formed in the embryonic stage. This notochord is formed on the which side, it seems? Dorsal side. During embryonic development, this dorsal side of the notochord is forming. If notochord is present, those animals are called chordates. If notochord is present, those animals are called chordates. Example, cephalochordates, urochordates, feces, mammalia. See, feces means fishes. Even fishes have also notochord. And mammals have the notochord. So these all are considered as chordates. If notochord is absent, what we called, if the notochord absent, they will be, or those animals are called non-chordates or porifera, or the non-chordates. Examples, porifera to hemichordata, porifera to hemichordata. So like this, notochord is present and absent. So what we discussed in this class, coelom, eucoelomates, pseudo-coelomates, notochord, segmentation, like that we discussed. So next one, classification. In the next class, what we are going to discuss, that is important. So next class we are going to discuss about, classification of kingdom Animalia. Classification of kingdom Animalia. Next class we will discuss. Okay children, thank you very much. Next class we will meet.